Once you have understood DNA structure, it is quite a bit easier to understand RNA structure as it is a lot simpler. So RNA stands for ribonucleic acid. And just like with DNA, deoxyribonucleic acid, it's named after the sugar that is found. So RNA is single-stranded molecule. DNA was a double-stranded. So this is one strand with the bases here. DNA had a second strand with the bases being complementary. RNA is single-stranded on the other hand. But RNA is still made up of nucleotides like DNA is, with a sugar phosphate backbone. So there'll be a phosphate there, a sugar there, where the base is attached, phosphate there, sugar there. But the big difference here is that in this case, the sugar is ribose. Then there will also be nitrogenous bases. So these are nitrogenous bases, these colorful parts over here. And the key shows you the names of them. Now, DNA has the nitrogenous bases adenine, guanine, cytosine, and thymine. But in RNA's case, there is no thymine as a nitrogenous base. Instead, thymine is replaced by uracil. So there is still adenine, guanine, and cytosine, but there is now uracil in place of thymine. So that is one big difference between DNA and RNA. So there are three types of RNA. They aren't too complicated. We name them after their job that they do in protein synthesis, which we will cover in another video. The three types are messenger RNA, transfer RNA, and ribosomal RNA. So mRNA, we just do a small m and RNA, small t RNA, is the RNA molecule that takes the DNA code from the nucleus to the ribosome to actually make the protein. Transfer RNA is the RNA that brings amino acids, and ribosomal RNA is RNA that is actually found inside a ribosome, which is an organelle in a cell. So it is important to note that although we show the structure of RNA as being quite simplified, with this straight line structure with the bases being very visible, a lot of the time the RNA structure can look quite different, and this is normally visible during the process of protein synthesis, because each of the different three types of RNA will have a different structure. So in this diagram, for example, this is the structure of messenger RNA or mRNA. Single-stranded, straight molecule is not coiled, that would be DNA, and that piece of mRNA has copied the code of the DNA molecule that codes for a protein, so it's like the recipe. Then that messenger RNA leaves the nucleus, and this structure here that looks like a figure of 8 / a snowman is a ribosome. So ribosomal RNA you can't actually see in a picture. But this structure here, where there's three little nitrogenous bases, 1, 2, 3, 1, 2, 3, those are transfer RNA molecules. And what you look for is these little loopy parts on the side. So it's a molecule that carries an amino acid. So there you can see that is carrying an amino acid. This one has already dropped off its amino acid. This one is currently adding its amino acid to a chain of amino acids. So these are the tRNA molecules that only ever have 3 bases, whereas the mRNA molecule has a lot more than 3. Even though this diagram shows that there are 9, there will be a lot more than 9 even. Looking at another image of tRNA, you can see these folded parts of the molecule very clearly. That would be one folded area, one folded area. But what is very important to notice is that we only ever see 3 nitrogenous bases on a tRNA molecule. So in this case, you have cytosine, uracil, and cytosine. That is 3 bases. So that means that we only see 3 nucleotides, because if there's 3 bases and each nucleotide has 1 base, that means there are 3 nucleotides. But we will deal a lot more with pictures like this. For now, I just wanted you to see the folded parts of the tRNA molecule. Again, you can see that it's carrying an amino acid, but it is very different to the straighter molecule that is mRNA.